Now we will be classifying genetic diseases in a way that we can understand them. Okay, so genetic diseases can be classified. Genetic diseases can be classified into three. Okay, the first classification will be single gene disorders. Single gene disorders. Single gene disorders. Okay, the next will be complex multigenic disorders. Complex multigenic disorders. And the last classification will be chromosomal disorders. Chromosomal disorders. Single gene disorders can further be broken down into those Mendelian that are Mendelian in nature, where their pattern of inheritance follows Mendelian laws, and those that are non-Mendelian. Those that are non-Mendelian. Complex multigenic disorders are simply disorders that involves a lot of genes and the environment. Okay, so there's an interplay between multiple genetic portions and of course the environment, and that's why it's called a complex multigenic disorder. Okay, examples of complex multigenic disorders include hypertension, obesity, diabetes, type 2 diabetes mellitus, okay, these are examples of complex multigenic disorders. Mendelian disorders can be classified as dominant, they can be dominant, or they can be what? They can be recessive. Okay, and this dominant can either be X-linked, or autosomal. The same applies to recessive. It can also be X-linked or autosomal. Okay, now X-linked disorders are disorders that affect the chromosome X, while autosomal disorders are disorders that affect other chromosomes. Okay, so chromosomal disorders can be structural disorders, or numerical disorders. Okay, now structural disorders, examples include the translocations, the inversions, ring chromosome, deletions, insertions, isochromosomes, okay, while numerical disorders are aneuploidies, and euploidies. Okay, and under aneuploidies we have the monosomies, trisomies, tetrasomies, etc. And then Under the euploidies, we have the triploidy, triploidies, tetraploidies, etc. So non-Mendelian disorders are majorly 4 disorders. There are 4 that are non-Mendelian. Okay, we have the genomic imprinting, we have mitochondrial genetic disorders, mitochondrial genetic disorders. We have the trinucleotide repeats, trinucleotide repeats, and then we have the mosaicisms, genetic mosaicisms. So these are generally the classifications of genetic diseases. So let us talk a little about them. Now, single gene disorders are disorders that affect just a single gene, okay? Just a single gene. And usually they have large defects, okay? So they are also called single gene disorders with large effects. And these single gene disorders with large effects have been classified based on their inheritance patterns. Some of them follow the classical Mendelian pattern of inheritance, and those ones are called classical single gene disorders. And those that follow the classical pattern of Mendelian disorders have now been divided into whether they are dominant or whether they are recessive. Of course, dominant diseases simply means that it is the dominant gene that is affected, while recessive diseases simply means that it is the recessive gene that is affected. What is a dominant gene? A dominant gene simply means the gene that most common or that manifests itself. That is what a dominant gene is. While a recessive gene is a gene that masks itself or that is masked, okay, by the dominant gene, okay. And these dominant diseases can either affect the X chromosome or it can affect autosomes. So we can have autosomal dominant diseases, we can have autosomal recessive diseases, we can have X-linked recessive diseases, and we can have X-linked dominant diseases. And these diseases have their different characteristics. Examples of autosomal dominant diseases include Marfan's syndrome. Okay, examples of autosomal recessive diseases include sickle cell anemia. Examples of X-linked recessive diseases include hemophilia A. Okay, and then examples of X-linked dominant diseases include Alport's syndrome. Okay, now when we come to the non-Mendelian single gene disorders, these are gene disorders that are not inherited by the classical pattern. Okay, so their pattern of inheritance is non-classical. Genomic imprinting is one of them, where certain diseases manifest even though the mutation is occurring in a particular gene. The manifestation is different depending on whether it is the paternal gene that is mutated or whether it is the maternal gene that is mutated. Examples of this condition include Prader-Willis syndrome and Angelman's syndrome. Okay, mitochondrial genetic diseases. There are certain genes inside the mitochondrion. Okay, and usually it is believed that the mitochondria is majorly inherited from the maternal from the mother. Okay, so mitochondrial genetic diseases are also diseases that do not follow the Mendelian pattern. Trinucleotide repeats, okay, are also diseases that do not follow the Mendelian pattern because the mutation actually involves repeats of a particular portion of the genome, of that gene, okay, and then of course mosaicisms, okay, inheritance of different forms of chromosomes in an individual. Okay, that is that about the non-Mendelian diseases. Of course, we've already talked about complex multigenic disorders, and then chromosomal disorders. Chromosomal disorders can be structural, okay, where the disorder is because of alteration in the structure of the chromosome. For example, in translocation, we've already seen what translocation is, okay. In inversion, one side of the chromosome twists, okay, and of course there's an inversion. So for example, let's assume that this is a chromosomal portion, okay, this is portion A and this is portion B. In inversion, what we have is portion B coming here and portion A coming here. So this is what is called an inversion, okay. Okay, we also have a ring chromosome where the chromosome will cyclize, and probably because of loss of the telomeres on these ends, these two ends will now fuse to form a ring chromosome. Okay, and then we have deletions, okay, where a portion of a chromosome is completely removed, is completely lost. Okay, and then we also have insertion, where a portion of a chromosome has an addition to it, okay, that is an insertion, okay. And then we have isochromosomes. Ideally, a chromosome is supposed to be like this, having a long head and a short portion, okay, and a long portion and of course a short portion. In isochromosome, the division that happens made it to be that the division, instead of happening at this plane, okay, instead of the division to happen at this plane, the division happened at this plane, so that both short arms moved away to the daughter cell, and then both long arms moved to another daughter cell. Okay, so that is what an isochromosome is. Okay, so there's just either a pair of long arms or a pair of short arms. That is what an isochromosome is. And then of course, chromosomal disorders can also be numerical. Okay, numerical disorders are simply an addition to the number of chromosomes in that cell. That means that the chromosome has an increased gene dose. And these numerical disorders can be divided into euploidies. Euploidy simply means that the chromosome number is in multiple of 23. Of course, remember that the human karyotype has 23 pairs of chromosomes. Okay, chromosomes. Okay, 23 pairs. So if it is a euploidy, it means that it is in multiple of 23. If it is an aneuploidy, it means that the number of chromosomes is not in multiple of 23. Okay, and that's why in aneuploidies we have monosomies, meaning that one chromosome does not have its pair in monosomy. So chromosome 17, for example, should be paired. There should be two alleles, but in monosomy, that the second allele is missing. So we have only one chromosome 17, for example, in monosomy of chromosome 17. And then we have trisomies, where there's an addition of a chromosome to a complementary pair or to its pair. And one classical example is a trisomy 21, what we call the Down's syndrome. Okay, so those are trisomies. And then we have tetrasomies also, where one chromosome, instead of having a single pair, has two pairs. Okay, so there are 4 chromosomes in that case. Now euploidies can be triploidy, okay, meaning that every chromosome in that cell has an addition. Okay, of course, that will bring us to about 69 chromosomes, and that is usually seen in partial mode. Okay, even tetrasomies can be seen in partial mode, where it takes us to about 96 chromosomes. Okay, so that is basically a summary of genetic diseases. I hope you enjoyed this video. Actually, this video was made on request. If you want us to make more videos like this for better understanding of certain courses in pathology, please drop your suggestion in the comment section. Thank you so much for watching till this point. I would like you to share this video, and if you have not subscribed, please subscribe.